Good morning, ladies and gentlemen. My name is Jatuporn Sujjitjoon. I'm delighted to be here and I would like to thank for your kind introduction. Today, I will talk to you about the basic techniques of genetic engineering technologies. I have to excuse if this topic is quite too easy for you all because I see our expertise in front of this room. Okay. I will explain to you in two sections. The first part, I will mention and explain about the basic techniques used in molecular cloning techniques. And the second part, I will give an example of our case study, how to design the fifth generation car and how to establish the fifth generation car in Sirat Hospital. Here is genetic engineering, more also called as genetic modification or genetic manipulation. The definition of genetic engineering here is described by the NIH as it is a process that uses laboratory-based technologies to alter the DNA makeup of an organism. This may involve the changing of nucleotide, single base pair, or sometimes deleting the region of the DNA, or adding a piece of the segment of DNA to improve the organism trait or phenotype. Here, nowadays, the genetic engineering technologies are very powerful tools to develop advanced biotechnology that help to improve our quality of life. For example, here, the technology can help to improve the yield and quality of the plants and animals for agriculture and can help to produce the desired biological molecules in an industrial biotechnology. And in the field of research, it can help us to learn how to cells and the gene function in the health and the disease. And in the field of medicine, it can develop the new treatment in the field for treat the patients. Today, I will focus on the technology that we use in my research field, that is cancer immunotherapy, especially CAR T cell therapy. The history of recombinant technology was established in 1972 by these two professors, Professor Boyle from University of California, who discovered a restriction enzyme that cut DNA strand, a specific DNA sequence that could stick to other pieces of DNA. Another key person of this technology is Professor Cohen from Stanford University. He discovered that the method to isolate genes in antibiotic-carrying plasmids and can propagate in E. coli bacteria. These two professors had also attended in the same meeting as we are here, and they discussed together and thinking about how to integrate these two knowledge for apply in the field of biotechnology. So they decide to do the big thing by using the plasmids can use as a vector for cloning individual DNA segments. And nowadays, he established a biotechnology company called Genetics and create the recombinant DNA or protein that's very powerful in this field. First of all, I will explain a little bit about the plasmids. What are plasmids? The plasmids are small DNA molecules within the cells that are separated from the chromosomal DNA of the bacteria. They are most commonly found in bacteria. It is a small, circular, double-stranded, naked DNA. And plasmids often carry genes that are essential for survival of the bacteria, such as genes for antibiotic resistance. And the plasmid itself can replicate within the host cells. In the field of molecular cloning, artificial plasmids are used as the vectors to drive the replication of recombinant DNA within host organism that we use nowadays. The component of plasmid that are normally cloned in normally used are the multiple cloning sites that you can insert the gene of interest within the plasmid, the promoter region that can drive the gene expression, the origin of replication or ORI, and another gene that is essential to select the clone, such as the antibiotic resistant gene. This slide shows just overall the workflow of molecular cloning. The technique will use the restriction enzyme to cut the plasmid backbone and insert DNA. After cutting the fragment of plasmid and the insert DNA, these two fragments will be ligated into the plasmid by using the enzyme called DNA ligase. After creating the recombinant plasmid, the plasmid will be transformed into the E. coli bacteria. And after that, the transformed colony will be isolated. And after plasmid were purified, the DNA sequences were confirmed by DNA sequencing. Here, in general, there are several techniques that using for molecular cloning. I give you just example of the technique that we use nowadays. That I list here in this slide. The first technique is using restriction enzyme cloning. This technique is the most popular technique nowadays. That our lab usually use this technique also to construct recombinant CAR in our lab. Another techniques such as gateway recombinant cloning, topo cloning, Gibson assembly cloning, and other options were available in the market nowadays. The differences between these techniques are based on the using of restriction enzyme or using specific enzymes and specific cloning vector. I will mention in the restriction enzyme cloning technique. This restriction enzyme cloning is the classic method that is the most popular nowadays. It is called restriction endonuclease. The enzyme can create double-stranded DNA at specific sequence sites in the DNA. It cleaves the DNA into the fragments. And they have the capacity to cut the DNA, so this enzyme also called molecular scissors. The other enzyme that is very useful in the technique is DNA ligase. The enzyme can join and bind the two DNA fragments and form phosphodiester bond. So the ligase is also called the molecular glue to link between two fragments. The picture in your right-hand side shows the brief protocol how to clone gene of interest into the bacterial plasmid. You have to have the insert DNA that franking with restriction sites of enzyme. After you digest the insert DNA and the plasmid with the same enzyme, and then put them together and incubated with the ligase enzyme, the next ligation the ligase will join these two fragments into recombinant plasmid. Here, another technique is just briefly explain about gateway cloning strategy. Is another technique. Sorry. That is very easy and versatile in high throughput manner. This technique uses the special recombinant sites franking between the gene of interest and require these two reaction in the reaction for gateway cloning. And the third is the topo cloning. is use the topoisomerase vector. Use the property of enzyme that will frank the plasmid with the thymine overhang here, and use the property of tag DNA polymerase enzyme that can cut and leave the overhang A here in the PCR product, and do perform the ligation reaction with the topo vector. So the benefit of this technique is they do not use restriction enzymes and ligase. Another example is Gibson assemble is very useful to assemble several fragments. At least 15 fragments you can ligate within the one reaction, and up to 100 base pair to 10 kilo base pair can perform. I will end up with the first part of the brief explanation about molecular cloning techniques, and I will focus on the work that we do in Sirat Hospital. As Dr. Tanyavi described you all about the characteristic of CD19 specific chimeric antigen receptor T cell therapy or CAR T cells. We already know that we have to construct the CAR gene in the vector that we want to use. Usually nowadays we use the viral vector, but later on the non-viral vector-based system are developed. The CAR construct, I think the key success in CAR T cell development is how to design the CAR gene and construct into the plasmid DNA. And after we construct, we need to introduce the CAR gene into the T cells and do the CAR T cell to function in killing the tumor. Here is the brief structure of chimeric antigen receptor or CAR. As Dr. Tanyavi mentioned about the basic structure of CAR, composed of three main domains. The first is ectodomain, which used for antigen recognition. The hinge and transmembrane domain, and the third part is endodomain or intracellular domain that used for T cell signaling. The selection of different parts in CAR affect the function of the CAR T cells. For example, we include different co-symmetry molecules within the intracellular domain of the CAR. The different co-symmetry domain may affect different function of CAR, such as some may promote in vivo persistence of CAR, some may promote cytokine production. This slide shows the brief US FDA approved 6 CAR T cell products nowadays. There are 6 for CD19 CAR T cell were approved by the US FDA, and 2 products targeting BCMA for multiple myeloma. I will focus on the prices or the cost of CAR T cell production. For example, for a single infusion, 1 dose here, the price is very expensive. So in our country, there are limited the access of CAR T cell to patients. Therefore, our group realized that we need to develop our CAR T cell products in Thailand. Here is the development of different CAR generations nowadays. The second generation CAR were only CAR that were approved by US FDA nowadays. Now, and different other CARs were developed. For example, the third generation CAR, this CAR construct incorporated 2 co-symmetry signaling domains within CAR structure, for example, CD28 and 41BB. And the clinical trials were done nowadays. Another version of CAR is described as 4th generation CAR or armor CAR. Different paper describe different structure of CAR Sometimes they call armor CAR T because not only the CAR construct, they also co-express other genes that help to promote CAR T cell function or inhibit other cells to suppress CAR T cell. For our hospital, we also developed the series of 4th generation CAR. Here, we call Sierra 4th generation CAR. In our CAR construct, we incorporated 3 co-stimulatory domains within the intracellular signaling domain. Here is the structure of our 4th generation CAR. We include the intracellular domain of CD28, CD137, CD27. We propose that this combination of 3 co-stimulatory domains will help the CAR T cell to function efficiently, especially promote the T cell survival and in vivo persistence in patients. Not only 4th generation CAR that were developed in the lab, we also focus on the improving CAR T cell function by fighting against other immunosuppressive cells or protein. One of the well-known protein that were shown to suppress CAR T cell function is the immune checkpoint protein. And the PD-L1 is an immune checkpoint protein that is highly expressed in the tumor cell and usually will bind to the PD-1 receptor on T cells and suppress the CAR T cell function. So to minimize this effect, we developed for 5th generation CAR by adding the property of CAR4 can be secrete anti-PD-L1 SAV into the and secrete out of the cell. The function of anti-PD-L1 SAV will block the PD-1 interaction and help restore CAR T cell function. We choose a anti-PD-L1 antibody clone atezolizumab antibody that were approved by the US FDA nowadays for use in non-small cell lung cancer and other lung cancer. This is our case study of 5th generation CAR that were published in 2022, and just this year, we create 5th generation CAR targeting CD19 and test the in vitro model that can kill the leukemia and lymphoma cancer cells very efficiently. And another paper, we also create 5th generation CAR targeting BCMA antigen that targeting multiple myeloma cell line, and we already proof of concept that it worked well in vitro to cure the cancer, and the property of this CAR T cell also promote T cell proliferation and can decrease the exhaustion of CAR T cell also. This is our development of CAR4 construct. As you know, in Thailand, we have a limited funding. So we use a very easy way to clone the CAR4 construct. At the beginning, we used the old-fashioned method like overlap extension PCR, but it doesn't work well with us, and we give some suggestion from our collaborator. You can do just simple way, just select the sequences and synthesize the gene fragment that link between the gene that you want. For example, you select the gene encoding the CD8 transmembrane and intracellular signaling of CD28, CD137, CD27, and link to the CD3 CTAR. And you just synthesize this fragment, use as a template for in the PCR reaction. You just an easy way for our collaborator to suggest us. So that's led us to successful in generating CAR4, and we also developed the different CAR4 generation targeting different antigen, different tumor antigen by selecting different SCLB fragment that usually derived from antibody clone that you use in the lab. The strategy that we use the cloning of CAR4 cassette gene into lentiviral transfer vector by using traditional method, the restriction enzyme cloning strategy. I will give example, just very easy way to how to retrieve the gene in the coding sequence that you want when you put in the gene fragment. So we have just retrieve the sequence of the gene that you want from the database and select only the region that you interest. And when you retrieve the coding regions of the gene, and you map the functional domain of the protein and select only the coding sequence that you want and put into the software or software that I usually use is the SnapGene tool. You can manipulate the cells. You can just link the fragment of the gene that you want, and you send the company to synthesize the gene fragment. Here, after you have the CAR construct within the lentiviral transfer vector, after that, the challenging task is how to retrieve the SCV sequence from the database, because it's very like a secret from the company. It's very hard to find that sequence sometimes, but usually we can get the sequences from the publication, but some paper describe the nucleotide sequence and amino acid sequence of that SCV. So sometimes we can retrieve the sequence from the patents that is available in several databases, such as the Google patent is the very easy way to retrieve the SCV. Other database, for example, World Intellectual Property Organization or WIPO, and other databases from European and Japan also. I give you just example of how to retrieve the atezolizumab monoclonal antibody clone. We get the sequence here, and normally they provide only amino acid sequence, but sometimes they have nucleotide sequences also. Another database that you can retrieve the antibody or drug that used nowadays, such as the KEGG database from Kyoto. Here's just example of I can retrieve the atezolizumab amino acid sequence of the heavy chain and light chain here, and select only the variable region of the heavy chain and variable region of the light chain. They can put them together via linker sequences, and you can get the SFV coding sequence from atezolizumab antibody. After that, you can put this coding sequence within the upstream of the CAR4 cassette, so you can get the fully construct of the 4th generation CAR targeting specific antigen. And another thing that we design is we need to co-express the CAR protein and secrete the anti-PD-1 SAV also within the same vector. So we decide to use the T2A peptide clone upstream of the anti-PD-1 SAV. This T2A peptide can help us to produce two proteins within the same CAR construct. Nowadays, there are 4 available sequences of P2A peptides that use in the lab as shown here. The property of this 2A peptide will help the protein translation that the ribosome will skip the skip the production of protein at the glycine and proline here. So you can get 2 proteins within the 1 vector. Another example of how to co-express the protein is to use the IRAS sequence or internal ribosome entry site elements within the construct. the limitation of this IRES sequence is the size of the IRES sequence is quite longer than the T2A peptide, so it may affect the lentiviral packaging capacity. So I select the using of T2A peptide in our construct. Here, just summarize to you all about the CAR design. You can use the basic techniques, restriction enzyme cloning technique to create CAR construct that we can apply in several ways. We can design the CAR, our CAR, to produce and secrete some protein, especially immune checkpoint protein, or may secrete some cytokines to finally help to improve CAR T cell function. So after we got the CAR construct, the later step for produce CAR T cell is to how to deliver the CAR into the T cell or NK cells. Normally we use a viral vector, but nowadays the non-viral vector is coming very fast nowadays. So the next section will be described by our next speaker to describe you about the delivery system, how to produce CAR T. So I would like to end up with this slide. I would like to thank all of Grant and TTCI, Thailand Hub of Talent in Cancer Immunotherapy and Committee, and our colleague. Okay, thank you.